Welcome to Lesson 9 of Module 5 Heredity. In this video, we're going to be looking at frequencies of different characteristics in a population, and we're going to look at how we can measure those. By doing that, we're going to be answering the two inquiry questions listed here. How can the genetic similarities and differences within and between species be compared? And can population genetic patterns be predicted with any accuracy? So again, lots of meta language in this video. The first one is polymorphic, and that's when a gene has different alleles in a population. Allele frequency is the rate at which alleles occur in a population, and within the population, this must add up to 1.0. A fixed allele has a frequency of 1.0, and when we're going to discuss genes in this video today and in some of the examples, we usually represent these alleles as P and Q. When we talk about a population, it's any group of interbreeding organisms. Gene flow is the movement of genes, and equilibrium is the high level of gene flow, resulting in allele frequencies that are very similar. So between different populations, there must be a high level of gene flow for these populations to be considered a single interbreeding population. If their genes do not flow between these populations, they may be considered separate gene pools or populations. However, if we focus on a single gene when we're examining these frequencies, this can be due to random chance. So we need a wider scope to make sure that we can make these predictions and statements accurately, which is why that we sample many different genes and undertake some really robust statistical analysis to ensure that we do make correct statements about populations and gene frequencies. If there are any significant differences, this usually indicates that something is restricting the flow of genes between species. Sometimes that is physical barriers, but there can be some other reasons why this happens. The Hardy-Weinberg equilibrium is a model that we can use to calculate allele frequencies for a stable population at equilibrium. So from our genetic crosses, we know that a heterozygous cross produces a 1 to 2 to 1 ratio. And due to this, we have the following formula that allows us to calculate allele frequencies and how the percentage of the population that can contribute that allele to the next generation. And that formula is p squared plus 2pq plus q squared equals 1. And we can see that they've done this example on the right hand side here. And we're going to look at those in more detail. So while we use this model to calculate allele frequencies in a population, there are some limitations that we need to consider when we're calculating these. We know that all alleles are mutating at the same rate, or we assume this, so that inheritance is not affected. We have to ensure that there is no migration into or out of the population. The limitations is that we have to know that generations do not overlap. Population is not affected by selection, so therefore evolution cannot be taking place for us to use this model. The population must be large enough to not be affected by random events, and all mating must be random, so individuals do not show any preference or selection for mates. We can use genetic markers as another way to calculate gene frequencies in a population, and genetic markers are any piece of DNA, a part of a gene or a part of an intron, which has a known location on a specific chromosome. They can act as records of change, and when we're comparing genetic markers, we need to ask ourselves, are these individuals the same or are they different at this particular gene locus? For a genetic marker to be ideal for us to use in these calculations, it is polymorphic within the population of interest, so it must show variation. It is usually a neutral marker, meaning that it's not currently under selection, and it needs to be easy and affordable to work with. So there are different types of genetic markers that fit that criteria listed on the previous slide, which we can use to examine the DNA sequence. Mitochondrial DNA is often used because it only has one allele due to be inherited maternally. A single DNA sequence from a useful genetic marker will have multiple points of difference in a population. And these genetic markers can be used to work out which individuals are more closely related to another. And we can see that's what they've done with this sequence here. So they're looking for the presence of the genetic marker and seeing what differences or what similarities are happening there. So another type of genetic marker is a single nucleotide polymorphism, otherwise known as SNPs. So pretend that there's a little I in there. And these are single base changes scattered throughout the genome across multiple chromosomes. It its points of variation can be examined simultaneously, usually thousands at a time, and it provides a snapshot of genetic variation. However, developing these SNPs can be technically challenging and expensive, and usually they only have 2 alleles per marker. Another type of genetic marker we can use are short tandem repeats or microsatellites, and each of these are examined for size differences, and these are proving to be much cheaper and easier than sequencing a large number of individuals. These short tandem repeats are non-coding pieces of DNA that contain a string of repeating nucleotides. So the number of repeats varies between individuals in a population, and it can also vary within an individual due to homologous pairs of chromosomes. So here we can see this short tandem repeat 1 and the short tandem repeat 2, and we can see the similarities and the differences in the numbers of these tandem repeats. And they do have 2 versions of those, which may vary in length. So this male here, his tandem repeats are the same. However, the other people in this diagram, their short tandem repeats are different in length. We can also do DNA sequencing and profiling, which in genetics testing, it detects specific alleles, mutations, genotypes or karyotypes that are associated with heritable traits, diseases, or predispositions to diseases. And we can do this by 3 tests. We can do molecular genetic testing, cytogenetic testing, or biochemical genetic testing. Molecular genetic testing is used to identify single genes or short lengths of DNA, and these can identify variations or mutations that in fact lead to a genetic disorder. To analyze these, they can be we can use tissue samples, and we only require a small sample to allow this to take place. And the 2 most common technologies used to undertake molecular genetic testing is PCR, so polymerase chain reaction, and then RFLP, restriction fragment length polymorphism. So PCR, the polymerase chain reaction, is a process which makes many copies of a piece of DNA, and we call this DNA amplification. So the DNA undergoes many sequences of heating and cooling and allows the number of copies of the DNA fragments to double. To allow this to happen, there needs to be primers at the end of each sequence, which helps to target the gene being amplified. So we don't want to target the entire sequence of DNA to amplify it. And it's a key tool for generating a DNA profile of short tandem repeats. So we can see here we've added the primers, and these bind to a specific section of the DNA, which allows us to then copy the length of the DNA that we want, and then it undergoes these heating and cooling to allow it to amplify, and we get more copies, and this process continues. Restriction fragment analysis can provide us with information about DNA sequences without actually having to sequence the DNA itself. We do use restriction enzymes, and these are enzymes that can recognize short sequences of bases, and then it cuts the DNA at that specific section. A DNA fingerprint, it's not the fingerprint that you might usually think of, but it's a unique DNA profile of DNA fragments from restriction enzymes. So then these undergo gel electrophoresis, and this is a process that allows different size molecules to separate within a agarose gel. So part 1 of this technology is to cut the DNA using restriction enzymes, and then it's to separate these fragments using gel electrophoresis. So we can see here that we place the DNA samples into these wells of the gel, and then what will happen over time is they will move up this gel via an electric current that allows them to move based on their size. So the smaller the fragment, the further it will travel in the gel, but we can see that there are many fragments because there were many restriction enzymes cutting the DNA into more than one piece. And then we can use this, so we have the mother and the father here, and then we can compare it to the child. So we're making sure these are used in some paternity test. DNA profiling is also used to distinguish one individual from another, and It uses the non-coding sections of DNA that we've already talked about. And it undertakes the process that we've looked at. So we have to extract the DNA, we have to amplify it via PCR. We then use the electrophoresis process. So the restriction enzymes cut it up, we put it through electrophoresis, and then we can compare these to match up some DNA profiles. However, there are some issues with this process in terms of privacy, whether they can be contaminated during the process, and then how these DNA is stored after testing. So here, this is, like I said previously, a paternity test. So we're seeing the child and we wanted to possible father 1, possible father 2. So their DNA profiles are taken and then we see where they match up. That concludes lesson 9. Make sure you tune in for lesson 10, which is the final lesson of module 5.